On my presentation. I'm Sylwia Szymanska, and today I'll be talking about word embeddings capture functions of low complexity regions, scientific literature analysis using a transformer-based language model. Low complexity regions, LCRs, are protein sequences characterized by low amino acid diversity. In times where the proteome was still poorly investigated, researchers regard LCRs as non-functional regions and ignored them. Nowadays we know that LCRs can play an important role in cells. They are involved in binding molecules and take part in the phase separation or aggregation. They can also be related to neurodegenerative diseases. Unfortunately, knowledge about LCRs has not been collected in the systematized way. The databases that are publicly available, we have databases that focus on whole proteins, for example, the intrinsically disordered proteins described in this prot. We can also find databases focusing on specific regions in these proteins, such as low complexity regions. We can find databases containing information on LCRs with a specific function, such as in the MULOS metadatabase, regions contributing to liquid-liquid phase separation. Or we can narrow down the group of LCRs to homopolymers and search for them in the proteome, as in the case of the HRAP database. Since there are so many databases bringing together information on LCRs and functions, will this project make any contribution to the development of the study of low complexity regions? Let's start with the fact that at the moment there is no published database containing all LCRs and their functional annotations. The only reliable source of information about them are publications. Manual analysis of publications is time-consuming and sensitive to human error, but in the age of algorithms for function and structure prediction, it has become essential for database users to have manual annotation information. The number of publications is increasingly day by day, so manual annotation is becoming more and more challenging. In this project, we will focus on extracting LCRs function information from scientific publications using a language model to help and speed up LCR analysis. To achieve our goal, we used a language model based on a transformer architecture. We used text as input to get embeddings representing each of the words using the BERT-based model, one of the most popular approaches. Embeddings are vectors that represent a given token numerically. In our case, they will have a length of 768. Let us now discuss the stages of the project step by step. To begin with, I extracted and manually annotated almost 9,000 publications, from which the title and abstract were used for further analysis. The positive collection included publication describing LCRs with one of the following functions: phase separation, aggregation, DNA or RNA binding. The collection was divided into train and test dataset, and additionally the negative cases were enriched with hard examples. The hard examples are publications unrelated to LCRs, but the similarity of the tokens of those describing LCRs, they are more challenging to classify correctly. Let's take a closer look at one of them. Here we have two tokens and the embeddings calculated for them. To a human, these tokens are completely different, but translating them into machine language, they are very close to each other. This is evidenced by the cosine similarity value measure of 0.918. This measure is in the range 0 to 1, where 1 means that the two embeddings overlap. Next step, I converted the text containing the title and abstract into tokens. The analysis was performed at the token level, and this means that in this case the tokens correspond to words. Next, we calculated the embeddings for each token. We got multiple embeddings for a single publication. So the next step was to apply dimensionality reduction to them, so each publication was represented by a single embedding. Unfortunately, as humans, we have limits to our perception, and our world is understandable to us if it is in 3D. So to better illustrate how embeddings look, we use the same model but different dimensionality reduction methods to plot it in 3D space. On the slide, we can see in 3D a training set of embeddings obtained using the same model, but drawn using three methods of dimensionality reduction. We can notice that the best effect can be seen with the use of PCA. This set is separated by creating something like ribbons, where they are distinguished on the base of the subject of the publication. In the case of max pooling and UMAP, we see that the sets overlap, which will have a negative impact of the classification, so we choose PCA as the best approach for further analysis. To evaluate the classification quality of our approach, we choose lead suggest as the baseline model. It is a system created by scientists from NCBI, which, based on a set of publications and extracting information from them, classifies a new set of articles. In our approach, we used random forest for classification. Both approaches were trained and tested on the same dataset. As we can see, our approach got better precision and F1 score, while lead suggest has a slightly higher recall. For a better understanding of the results, let's take a closer look at two elements. In our approach, upload showing the classified publications from the test set and the lead suggest word cloud. Starting with our approach and the 3D graph, we see that there is a region in the graph where you can say all the ribbons come out. This area is extremely difficult to distinguish because there is an accumulation of both positive and negative cases. In addition, in the region of positive publications, we saw that there are single negative cases in the training set. These are publications containing hard cases. Analyzing the lead suggest word cloud, we can see that the algorithm has learned to recognize functions more. The lower precision is probably due to the fact that there is no aggregation or phase separation in the cloud. What's more, apart from the word poly, there is no mention of the name of the LCRs, so there were too unrepresentative for the algorithm to correctly recognize this publication. In conclusion, we created a model that classifies publications describing LCR functions based on information extracted from the text. Thus, we showed that using the language model, we are able to encode information about LCR and its function in a single embedding. This analysis was carried out using only free text consisting of the title and abstract, but I hope that the current trends regarding full open access to publications will contribute to increasing the popularity of this approach. And scientific text in the near future will also be kept in the form of embeddings, which will make the work of the scientific community easier. Thanks to my group for all the advice and support during this project. And thank you also for the invitation and the opportunity to present my work to a group of specialists.